Hello, everyone. I think we are about to restart. So my name is Anqi Qiu, and I'm going to present the work with my research fellow, Brian Qiu. So we're going to continue to talk about confounding factors. And I hope I'm going to give you some kind of computational solutions for that. And hopefully you can take this with you, and you can use some of the tools we introduced today. And I have very, very sexy title, and that Brian gave it to me. Actually, I didn't, Ryan gave it to me, so it's actually not what I initially think about it, but I guess that's much more sexier. Okay, so let's start. So compared to like situation 10 years ago, I think now we are in a much, much better situation in the sense that there are a lot of data available, and you pretty much can look for the data and to answer your research interest, and which is pretty good. And I'm going to re-show this picture that Tanya already showed from the biobank data. And basically, you have tons of phenotypes, and you will be able to link with multimodality imaging data and looking for the associations between the phenotypical data as well as with imaging data. And certainly, Miller basically shows a lot of potentials. Even with a subset of biobank data, we are able to define a set of imaging markers that actually can be markers for early disease detection, understanding. And certainly, if we go in beyond the phenotype, and if we look at the genetics data, and especially looking at the individual SNPs, and we can also look at the association between the SNPs with individual structures, such as subcortical walling. And the paper that published by Paul Thompson's group a few years ago, that already shows the strength of combining imaging data as well as genetic data to looking for biological markers that are related to brain structures. And certainly more aggressively, we also can link individual SNPs with individual voxels in the brain. So there are tons of association analysis we need to do in order to be able to detect real signals out of it. And certainly, there are a lot of challenges down here. But the question is, how do we examine massive these associations? And how do we boost statistical power for this analysis? And recently, Smith and Tom Nichols basically says even, at current stage, we have a lot of machine learning approach to deal with large dimensional data. But unfortunately, and currently, the workhorse tools of neuroimaging are still a massive univariate analysis, where a regression model is fit separately at each voxel. So that's still something we do at current stage. And what does it mean? So you have imaging data, let's call it Y, and you have n number of subjects, and Y basically is n number of subjects, and you have n number of voxels down there. And you have your phenotype data and genetic data, you write it down as M. That's the variables you are interested in. All the variables that Anne-Marie called exposure variables you want to explore. And certainly you want to look at association between Y and M. And the simple way we do is using regression models for each voxels. And you basically have a set of X. The X down here, you can see is confounding factors. And here I call it as the covariance of non-interest. Means that you put into your model, but you're not going to be interested in looking at what interpretation for this X. You're going to just be sure that you are fine with it. So you just put in there. And what you are really interested in is looking at beta M for your interpretation. And beta M can be easily written up because of linear regression, and you can simply have your analytic solutions. So for now, I just write down equation down here. But you look at your beta M and ask what are the statistical test results about your beta M. and in order to interpret the associations you are looking into. And certainly, there are many, many different things. I actually think the slides wasn't updated. This is the older one. But anyway. So you have your linear regression model, and you have your X, which is confounding factors. Just continue with the word that I'm going to talk about. And M is the variables we are interested in looking at, association between M and Y, and E down here is residuals, what we have. And certainly, under all this estimation and beta and so on, so we always have assumption by saying our observation regarding individual voxels, they has to be independent. And certainly, the variance of that has to be equal. Okay, and cross-individual subjects, they are also independent observations. So then you can make your statistical testing based on T values or F values. And you have your underlying theoretical, analytic T distribution or F distribution that allow you to determine the P values. Is it all true, right? So first of all, we have to think about the model, the linear regression model we assume is true. What does it mean? So X, we include enough X into the model. So that means your mean estimation is covered enough, okay? And of course, your E has to be, as what I just mentioned, and sigma I means your observations has to be independent to each other. And that's very, very challenging for imaging data because whatever we know about X is based on our prior knowledge. So whatever we know, for instance, we know pathology and inferences imaging markers, and imaging markers is related to depression. So you have this kind of relationship you know, so you have to put X into it as your confounding factors, okay? But if you really model every confounding factors, and certainly the residuals we are looking at will be perfect estimates, right? So that means you don't have inflation on your residuals if you don't include X into your model, okay? And of course, we know when the residual is inflated, and we can't lose the statistical power for detecting real effects, okay? That's how important to put the X, the covariates or confounding factors into your linear regression model, okay? And even beyond that, there may be some variables, unknown variables. It's just that we never think about. From prior knowledge, we never know this variable, we call it Z, that could be inferences Y, but in the meantime may also have some association with the variables you are interested in down here, which is I written down in the other box. So as Anne-Marie mentioned, if you miss that Z variable into your model, and your estimation for M can be biased. It can be biased to positive side or negative side. And so we don't really know about that. But in addition, beyond what Anne-Marie talked about, these Z variables we actually really don't know because we never measure it. Probably at the beginning of the study design, we never really think about to collect such data and for your particular study. So you basically don't really understand, what this variable can be. So how do we do with that? Why don't we just put it into the linear regression model? So that means we have unknown confounding variables Z in the regression model. Then I also have the coefficients that associate with this Z. Okay, so I'm going to put entire these things into regression model, and certainly I don't know beta X, I don't know beta M, and I don't know Z and beta Z as well as the residual, the errors we need to know. Okay, so this is kind of somewhat like a linear regression model with things you know and plus the things you don't know. It's kind of a mixture of both together. And of course, the aim we try to achieve is that to get a best estimate for beta M in order to make much robust statistical inferences on beta M. And that's really what we want to do. And certainly for this kind of setting, for this linear regression, so we need to solve a set of variables, right? As I mentioned, beta x, beta m, z, and beta z, as well as the variance of E. And certainly, there's another variable is hidden in the regression model, which is number of z's, number of missing confounding factors we need to add into it. Okay, and that's the entire thing we need to estimate. I'm going to briefly show you technically how we will estimate those things, and hopefully you get an idea, even though you may not in the mathematical, technical way to try to solve yourself. So now I'm going to say how we will be able to do statistical testing for those things, and hopefully I will be able to convince you that those approaches can also relax the assumptions for the residuals, in the sense that residual cross the spatial domain in the imaging domain, so they can be correlated with each other, and cross individual subjects, they can also have some relationships. So those methodology will be able to allow you to relax a lot on the assumption of linear regression. So how do we do? Certainly, there's some very beautiful mathematical tools called the QR decomposition in math. And so they basically allow you to decompose a matrix to be a multiplication of orthogonal matrix and times the upper triangle matrix. This upper triangle matrix is very important because that makes your linear equation simpler. So you can basically divide the first top linear equation into 3 linear equations, while the last linear equation you only have unknown variables of z and beta z. Okay? And basically the remaining m and x will be in the first 2 equations. And that's very nice, right? Because we can solve in the backwards, so from the last equation and to the second equation and to the first equation. Okay. So let's just look at the first equation down here. And certainly if you look at the equation in the middle down here, and this much look like you have y, which is your imaging data, and certainly QZ you already know because you do a QZ decomposition for the x and m. So this can be easily, these things is already known. And of course, since you don't know is z beta, and z and beta z, as well as the residual terms. Okay, and this is very much like decomposition, matrix decomposition problem, such as you can use principal components analysis, you can use independent components analysis, and so on and so forth. So you can use many different approaches to have estimates for z and beta z. So here, what we prefer is to do a factor analysis because they can count the spatial correlation among your neuroimaging data. And also, they can count the quite number of spatial, just cross subjects, and there's, the correlations across individual subjects. And furthermore, what we have is how many z's you're going to choose. And certainly, there's many different approaches you can do. So the best way we test it is using the eigenvalue ratio approach. So that means based on how fast the eigenvalues for y is dropping, and you basically can determine how many z values you can choose. And just give you a brief write-up, and you don't really need to remember all these equations. And based on this, principal components analysis, we can just do a single value decomposition, then you can easily get your solutions for z and beta z. Okay. So now we know what z and beta z is, and we just need to go one step back, and by saying how we will be able to solve the second equation, because beta m is really the one we are interested in, right? So what we do is that we not just think about z is something independent of x and m. Beta z is actually something relative to the x and m because we call it as confounding factors. Confounding factors that must have some relationship with m as well. So what we do is that we assume z is a combination of x and m, which we know, and of course alpha x and alpha m is something we don't know, and w here is y naught is all the residuals we think about. And Z is the one we already estimate from the third equation. So you basically have some sense on what it's going to look like. Then we will be able to use this equation and plug into the second equation down here, and you pretty much get the equation down here. So all these things you have is relative to M, and you have beta M down here. And beta M and alpha M star is the one that basically is the unknown parameters we need to estimate down here. And again, that's become much simpler because this is a pure linear equation problems. You can use robust regression or sparse regression, approach to really estimate the beta M from it. Okay, and just give you a brief overview how we will be able to estimate those parameters. So first, we will do a QR decomposition for the matrix of X and M. So we'll be able to get in 3 equations down here. And we basically solve these 3 equations by first solving this one and that one. And probably we don't need to solve this one because there's nothing. We already estimated our, the interest, the variables we are interested in. So basically, we just leave the first equation down here. Otherwise, again, that's just become a pure linear equation. So you can use robust regression to solve that equation. So now, once you have beta M and you know what your residual is, then simple thing you can do, we can compute this, we call the wall score, and for parametric hypothesis testing on beta M at each voxel. And smaller v down here, it just indicates as which voxel it is, okay? And C down here is a contrast matrix, depends on how you want to do the combination of M and how you're going to do the statistical testing for it. And TV down here, you have distribution associated with it, which is approximately it follows the distribution for chi-square with a certain degrees of freedom. So it's that wise approach to do a statistical testing based on this statistical model. Answer may not be true, right? Because we know parametric, maybe you have underlying distribution down here, and normally this distribution follows certain assumptions. So what we normally do is using a non-parametric approach instead of parametric approach in order to be able to relax the assumptions for residuals and so on and so forth. So what do we do with it? So the way we do it is normally people say, oh, I just do permutation tests. And the permutation, the way you permutate your data is you permutate your subject's variables in the sense that I permute my X and M simultaneously and then use the linear regression against the image measures. Is that true? This is the correct way to do it. The answer is no. Because you have confounding factors X and Z down there, you can't easily permute it in that way to get the permutation done. The wise way to do it is define a permutation matrices. Down here, I written as FBR. And you permute the residuals of your linear regression model, the model we just mentioned, okay? So you have to permute your residuals over there. And certainly, we are a little bit complicated because we have rotation, this Q, rotation matrix in front. And certainly, one way you can do is permute on the residual, the rotated residual, rotated by this Q we introduce, the orthogonal matrix we introduce. And this is what we write here. We indicate as FBB is permutation, and R down here is indicates the rotations down here. But certainly you can do other ways of permutation because permutation still requires the independence of, exchangeability of these residuals. If residuals is not exchangeable, then you can do a permutation test. The other way to overcome with it, you can use, for example, white bootstrap. Okay, so here FB down here can be representing a permutation matrix or can be a white bootstrap matrices. Okay, and certainly the other way to do the permutation, you still permute the residuals, but instead of permute rotated residuals, you permute non-rotated residuals. And people may ask me, what's the difference between the two? The answer is, initial our assumption is really on the residue itself rather than rotated residue. So that means if you rotate it, even this rotation matrix is orthogonal matrix. Ideally, theoretically, they could remain the exchangeability, but it's not necessarily true always, okay? So it's wise to think about to permute on the non-rotated residues, okay, to give you a more proper theoretical way to do it, okay? So now you have a way to permute data, and you just need to generate new data based on this permutation, and you rerun the model as I mentioned before. So you can rerun it, and you will be able to generate in each round, in each iteration, and you will be able to generate an image map that has these p-values, or the t-v values I just mentioned, the word scores I mentioned before. And what you do is, from this iteration, you find the maximum value from it, and you save it. As you repeat for 1,000 times, you will have 1,000 value of that. And you will be able to generate an empirical distribution for your statistics you are looking for. Okay? Once you have that, and what you do is at each voxel from your true model, you compare, you look at the location of this value is in this distribution, then you count the proportion from that. And that's your real p-values. It's corrected across the entire images. It's corrected for the entire image. And this is a normal way what people call it as family-wise error rate. Okay, family-wise error rate. But of course, this is a non-parametrical way of correction for the multiple comparisons across the entire images. Okay. So I hope I gave you a clear idea how this approach going and how you will be able to make a statistical inference on that. So you may ask me, is it really, really useful to embed these missing confounding factors in your linear regression model? And also compare non-parametric versus parametric. Is that necessary to do a non-parametric approach? Because computation-wise, for non-parametric approach, it's much more expensive than the parametric approach. So what I'm going to show you is to using simulation data and to basically demonstrate it's going to be really, really useful. If you really have missing confounding factors, it's good for you to embed it into it. So you estimate. And certainly, now non-parametric approaches sometimes can really boost a statistical power. How are we going to do that? Of course, the simulation, you always have to generate the data, the true solution, and you will be able to do it, right? But remember, so the basic equation we have down here is this linear equation down here. And we assume there are two missing covariants we don't know. And the sample size down here, we relatively use small sample size rather than large sample size. And people may ask me, why do you choose 114 subjects? And that's tied with our real, data, the number of subjects we have for that. And certainly, there are many, many different ways of generate, residuals. We call it noise distribution. So then certainly, there are many different ways to do that. So we basically gave us 16 scenarios. And the way you look at, the simulation results is to looking at this false positive discovery rate is pretty much around the level of 5%. And that's what you are looking at. And here, just give you some summary of noise distribution. You look at the spatial domain, cross subjects, as well as the spatial domain where the correlation are similar to each other. Okay, I'm not going to talk each line of this, but just give you some idea how many simulations we have done through for the experiments. First, let me show if you don't model the missing covariance, how much power you lose. Okay. If the confounding factors, the missing confounding factors have no relationship with our variable of interest that's showing in the first row down here, you're probably still okay without modeling, okay? But once the correlation between the confounding factors as well as the variables we are interested in become larger and larger, And from here is from correlation from 0.1 to 0.3. And you can see you basically, none of this approach, regardless whether it's parametric or non-parametric approach, you basically just, your false discovery rate is just going to be really killing. Okay? And in the second way, so when we look at how we will be able to do hypothesis testing, whether we should use a parametric approach or non-parametric approach, even for non-parametric approach, whether we should permute on the non-rotated matrix or rotated matrix. Here is the graph I show you down here. Hope I can convince you. And compare parametric versus non-parametric. In a lot of scenario, basically, Y bootstrap permutation will give you much, much better results. While if you look at the non-parametric approach in different scenarios, and you basically see we gain a lot of things when the spatial correlation is very strong. Okay? Which is the case in neuroimaging data, especially when the imaging spatial information, spatial correlation among them, at standard, the error variance is not equal as well. So those Y bootstrap on the non-rotated matrices will be much robust approach for that. Okay. So now I gave you some idea of if you have one phenotype of interest and you have your imaging data, and you will be able to use the approach to estimate missing covariance. So what about for example, for biobank data, you have tons of phenotype data you also want to associate with your imaging measures, or you have genetic or epigenetic data, which you have your M is very large dimension. How are you going to do with it, right? Because we need to do a multiple comparison, not just across the spatial domain, we also need to do a multiple comparisons across entire epigenetic data or the phenotype data as well as the genetic data. So what we're going to do is it. So I'm going to give you a hands-on kind of experiment and how you will be able to do this approach using a software package we create in MATLAB. And maybe some people warn me that everyone may not have MATLAB license. So just give you a brief overview. We're going to make executables available so that you will be able to use the executables and build into Python and call the functions so that you will still be able to run it without MATLAB license. Okay? And just give you an example, you can go to my website and you will see 2 links. Then you will be able to download the software as well as demo data you can use for this software, okay? So just give you some more hint on that. So what you first do is to prepare your data. What we have Y, which is imaging data. You make it into matrices and number of subjects versus number of voxels. We always do in that way, right? And you have your X, which is the confounding factors you already measured. So you can define it as well. So this becomes a matrix as well. N number of subjects versus N number of confounding factors. And CPG here, I use epigenetic data as an example. So I write it as a CPG. But you can think for any variables, I call it M. Okay? And same way, you can make it into a matrix as N number of subjects times N number of CPGs or N number of phenotypes you have. And this is just for preparation of your data. Then you are ready to go. So how do we do? Remember, we need to estimate the number of covariates. But if you just have one phenotype, M, and you are all set, you just need to estimate once, you go and you're perfectly fine. But once you have a lot of phenotypes, and depends on whether you want to, individual has N number of missing covariates, or you want to have everybody just using same number of missing covariates. It depends on your own purpose. That's why we have separate modules for estimation of missing covariates. So you can take it out as the way you want to do, okay? So for this case, what I do is I basically assume all the CPGs against the imaging measures, they have same number of confounding factors, okay? So that's why I do what I do. I have this small little loop down here. This is n number of CPGs. I estimate it. I recorded down the number of missing covariance across, for example, 100 CPGs. And I basically say what is the common number of missing covariance across these 100 CPGs. You can use that as your model and put that number into your next step, okay? Then the next step, basically, we need to think about if we want to choose a non-parametric approach, we need to compute this resampling matrix, this R matrix I just mentioned to you, right? So this F matrix I just mentioned to you, well, for permutation or Y bootstrap. So for this case, I basically say I'm going to do 1,000 bootstraps, okay? And you will be able to use this function down here to just generate that F matrices for your purposes. And the next step, what we're going to do is to compute the real model, right? If I use wild bootstrap for this, with the family-wise, error rate correction, then you have to really start the wheel and to start to run it. So how do we do it? So you see we have two major loops. One, the first loop is about your phenotypes. Let's say we have 100 CPGs down here. That will be our first loop. The second loop down here is number of permutation you do, okay? So down here, before you start your resampling stuff, what you do down here, you just use your real data to estimate your parameters, your beta parameters you need to do, okay? Your beta parameters you need to do. And down here, in the resampling one, you just need to plug in your resampling matrix, generate new data, and refit your data, and get the statistics out of this loop, okay? And certainly, you have to do something afterwards, which is, to correct for multiple comparisons. And what we do down here in terms of code, and there's one single line that does all this two-loop computation for you. As long as you define your Y variables, your X variables, your CPG, and number of missing covariance you need to do, and resampling matrices, and you are ready to go, okay? So now, how do we compute the corrected p-value for each voxels? And same thing, the same equation applies. In this case, if you resampling your data for 1,000 times, you will be able to generate, empirical distribution from these 1,000 scores. And you compare your the statistical scores at each voxel with these scores, regardless of which CPG it comes from. You have to do this for every CPGs, okay? So now, what is the technical questions I'm going to ask you? So is the same resampling matrix, is the resampling matrix the same across all the CPGs? Yes or no? No? And anybody says yes? It's actually yes. You need to have the resampling matrices the same for every phenotypes. Otherwise, how them can be comparable, right? So that's why we define the resampling rate in the third step before the complicated, equation. So that's number one thing. So did you realize how do we correct for multiple comparisons across the whole brain as well as across all the CPGs, all the phenotypes? Did you realize how do we do that? Based on the family-wise error rate. Anyone knows how to do that? That's correct. So basically, for each resampling, each permutation you do, resampling you do, even though it's in the loop of CPGs, but you first look for the largest value across all voxels, but you also have to look for the largest voxels across all CPGs. That's why when your resampling is 1,000 times, your maximum value of that across all CPGs and across all spatial domains, it still gives you 1,000 numbers for generating empirical distribution for statistics. Okay? And that's how you achieve the multiple comparisons across the spatial domain as well as across the CPGs. Okay, perfect. So again, this is I just mentioned to you, so you basically have this comparison down here. So we basically use this approach to actually explore the ICP genetic markers in association with neonatal imaging data. And our sample size is relatively small, it's only about 114. If we use a regular approach like GWAS approach and EWAS approach, there's no way you can get any statistical significance out of it. But what we found down here, we have CPG markers that actually associate with these genes, and particularly related to our calcium binding in the thalamus region. And everybody knows the thalamus region is pretty much the iron and calcium concentration location. So we'll be able to see this clearly at the neonatal stage. Okay. So I think this is demonstrating using a real data, and using this approach, we are able to actually boost the statistical power. But you may want to ask me how many covariates you estimate from this approach. For this one, we actually have 2 missing covariates we estimate from the entire data. And apparently, one of them is related to the brain size. Because we are looking at diffusion tensor data, we didn't say we need to put the total brain size as a missing covariance. So once we, estimate the missing covariance, we try to figure out whether any variables we collected is something to do with these variables. When we link with in our database, we found that this missing covariance is actually highly relevant to the total brain size. So this is something that learned from the model. We actually now need to think about even diffusion tensor imaging data. We probably wise to put a total brain size into your statistical model. Okay. To just give you a summary, and today I basically follow the previous 2 presenters and talk about confounding factors, and hopefully I give you a solution that you will be able to handle large dimensional data as well as these missing confounding factors. And I also give you very practical approach for non-parametric testing, so that in order to improve statistical power and control for a false positive rate as well. And there's missing slides, I guess, from here, but I just want to mention down here, you see my MATLAB code is actually separated into modules. Just give you some hint. If you say, I'm actually not technical person, and it's just going to be hard for me to actually really code those things and so on and so forth. What you can get from this, for example, if you already have your data and you just want to know whether you have missing covariance, what you do is go back to the second step down here in this step. So you can call this a specific function, just test your linear regression model whether you have already incorporated good confounding factors in your model. And this outcome value down here, if it's 0, that means you're perfectly set. If you have other things going on there, and that means you probably have missing something you think in your mind and see whether you want to estimate it. The second thing you can get from this is like the modules for generating resampling matrices. And down here, we pretty much provide 2 approaches. One is permutation. The other one is Y bootstrap. So that means you can use any way you want to. So that means you have your regression model. You take these 2 matrices, you can immediately permute your data wisely. Okay? And that's something beyond what is the entire flow really is, okay? And of course, a more complicated way will be calling the first step to estimate the entire model. But I guess that depends on how big your phenotype data is. If your phenotype data is not that large, it's pretty fast. 1 second you get 1 second you get your results. But if your phenotype data is really large, then you have to think about parallel computing and putting into a cloud or something, to do the estimation. And with that, I end my talk.